Geneticin, also known as G-418 sulfate, is a broad-spectrum antibiotic commonly used in molecular biology and biotechnology research. It is derived from the bacterium Micromonospora, rhodorangea, and exhibits powerful inhibitory effects on the growth of bacteria, yeast, and higher eukaryotic cells. The mechanism of action of geneticin involves disrupting protein synthesis in cells. It specifically targets the ribosomes, which are cellular organelles responsible for translating genetic information into proteins. Geneticin binds to the ribosomes and interferes with the elongation of the protein chain, ultimately leading to the inhibition of protein synthesis. This disruption of cellular processes prevents the survival and growth of cells. In molecular biology, geneticin is widely used as a selectable marker in cell culture experiments. It serves as a tool to identify and select cells that have successfully incorporated a foreign gene of interest. This is achieved by introducing a plasmid or viral vector containing the gene into the cells and adding geneticin to the growth medium. Only cells that have taken up the genetic material and express the resistant gene can survive and grow in the presence of geneticin. By employing geneticin as a selective agent, researchers can effectively propagate and isolate cells that have acquired a desired genetic modification. This could involve the introduction of recombinant DNA, gene editing techniques like CRISPR-Cas9, or the knockout of specific genes. Geneticin's selective pressure ensures that only the desired cells continue to grow, allowing researchers to study the effects of the genetic modification in a controlled manner. Geneticin is commercially available as a powder or as a solution, typically as G-418 sulfate. The appropriate concentration of geneticin in the growth medium varies depending on the cell type and experimental requirements. It is common to use concentrations in the range of 100 to 800 ml. However, the optimal concentration should be determined empirically for each cell line to balance effective selection with minimal toxicity or interference with cell growth. Generally, most cell types tolerate geneticin well, but it is essential to consider the potential toxicity. Prolonged exposure to geneticin can lead to the development of resistance in some cell lines, requiring higher concentrations or alternative antibiotics for continued selection pressure. In summary, geneticin, or G-418 sulfate, is a widely used antibiotic in molecular biology and biotechnology research. It inhibits protein synthesis by targeting the ribosomes, making it an effective selective agent in cell culture experiments for propagating cells with specific genetic modifications. By incorporating geneticin resistance genes into cells, researchers can identify and study cells that have successfully incorporated the desired genes, enabling a deeper understanding of gene function and facilitating genetic engineering applications.